Welcome to this explainer. Today we're going to unpack a massive, kind of hidden complexity in medical research and the upcoming technological revolution that is poised to change how we develop life-saving treatments forever.
So, let's dive right into this medical mystery. How is it that after, 10 to 15 years of grueling research and pouring up to $2 billion into development, the vast majority of our most promising drug candidates just stop working?
Well, the sources reveal a pretty staggering statistic. An astonishing 95% of pharmaceuticals that actually succeed in preclinical animal testing ultimately fail when they reach human clinical trials. Think about that for a second. That means for every 100 drugs that cure or safely treat a lab animal, 95 of them do absolutely nothing, or worse, cause unpredictable harm when they're finally given to a human being.
But here's the catch. The hidden reality behind that data is a systemic publication bias that heavily favors positive findings. Journals are super eager to publish when a drug cures a disease in a rat, right? But they routinely ignore the studies that produce negative or just inconclusive results. It basically leaves the broader scientific community with this distorted, overly optimistic illusion of the whole animal testing enterprise.
Let's look at Section 1, the biology, the extrapolation gap. Our first major roadblock is what's called trans-species modeling theory. Basically, this scientific principle explains that because humans and mice have completely different evolutionary adaptations, we simply aren't wired the same way when it comes to complex biological responses. We're literally looking at two entirely different complex systems.
The data brilliantly illustrates the metabolic gap. Cytochrome P450 enzymes, which are the specific enzymes controlling how drugs are metabolized in the liver, they vary so significantly across species families that an antibiotic perfectly safely processed by a dog might actually cause fatal liver toxicity in a human. And we've seen exactly this. There was a drug called trovafloxacin that was approved, but later had to be withdrawn due to unpredictable human liver failures that the animal models completely missed.
We see this extrapolation gap super clearly in a massive 10-year study on sepsis and trauma. Researchers were absolutely shocked to find that while humans shared uniform genetic responses to acute infections, the equivalent genetic changes in mice were, well, close to random. We thought we were looking in a mirror to predict human response, but biologically, it was closer to looking at static.
Moving on to Section 2, the methodology, the standardization paradox. Because it isn't just the biology, right? We also have to look closely at how the rigid methodology of laboratory environments actually ruins the data. A finding from a Purdue University study flips traditional science completely on its head. It proves that trying to perfectly standardize identical mice in identical sterile cages actually creates false positive results. We don't live in sterile, standardized bubbles, so why should our test subjects if we want to accurately predict how a drug will work out in the real world?
It turns out that hidden variables we might not even consider can completely confound a multi-million-dollar study. The sheer stress of small cages, bright lights, a lack of environmental enrichment, or even just failing to blind a study can completely skew the results. Get this. Here's an amazing fact from the research. Just the scent of male experimenters elevates a rodent's cortisol levels, physically dampening their pain response. You could think your new painkiller is a total game changer, when in reality, the mouse is just stressed out by the guy in the lab coat.
Which brings us to Section 3, the ethical pivot, the paradox of acceptable exploitation.
These persistent scientific and methodological failures naturally bring researchers and ethicists to a pretty profound impasse regarding the continued use of sentient animals. The literature impartially explores two distinct frameworks to navigate this. First, there's the current approach, utilitarian welfarism, which tries to minimize laboratory pain and improve conditions, but ultimately maintains the exploitation for perceived human benefit. Conversely, a rights-based paradigm provides a robust critique. It focuses on the moral status of sentience itself, arguing that sentient beings just shouldn't be instrumentalized at all, especially given how low the predictive success is for human medicine.
Finally, let's look at Section 4, the solution, a human-centered future.
Fortunately, we really don't have to choose between human health and ethics because a whole new technological paradigm is emerging to replace the old gold standard. To successfully overhaul drug discovery, the scientific consensus points to these three pillars. First, embracing new technologies called new approach methodologies. Second, updating the rigid regulations and funding models to support these innovations. And third, fundamentally changing the culture of biomedical education to champion ethical responsibility. At the very heart of this are organs on chips. These things are absolutely incredible.
They're tiny, basically USB-sized dynamic microfluidic devices containing real living human cells. They can perfectly simulate our lungs breathing or our hearts beating. Because they use actual human tissue, they capture uniquely human toxicities and metabolic reactions that animal models simply miss.
And regulators are finally catching up to the science. The 2023 FDA Modernization Act marks a massive turning point by officially allowing these advanced, human-relevant technologies, like organs on chips and AI in silico modeling, to replace animal testing in safety trials. It's a huge, huge step toward more predictive science. So, as we stand on the brink of this personalized, highly predictive medical future, I'll leave you with this provocative question. Are we finally ready to stop treating humans like lab mice and actually design medicine for humans? Think about that. Thank you so much for joining me for this explainer, and I'll catch you on the next one.